Hello, everybody. Hello, everybody. Can you hear me okay? I hope so.
Welcome. Hi, Ewan. I can hear you. Thank you. Paul.
Good. Great. Well, welcome back from your break. We're about to start the final third session. Lee, thanks very much for your kind remarks at the end of the last session. Your check is in the post. So this is to address this final session, the science that underpins the conservation of roseate terns. And we're going to have three presentations from Adam Seward, Chris Redfern, and Richard Berridge. And Adam, our first presentation today, I'll just say for anyone who's just, before we start, anyone who's just joined this session for the first time today, the questions and answers will be, box will be live as usual, so you can pop in questions as we go along. All of these three presentations are going to be live, not pre-recorded, so good luck to the three presenters, and thank you for doing that at relatively short notice, because they were all pre-recorded. So Adam is going to talk about the metapopulation dynamics of roseate terns in the UK and Ireland. And just a brief background on Adam. He's a conservation scientist and a nature photographer. He studied climate change primatology. He's worked and studied in diverse parts of the world, including Cambodia, Vietnam, Tanzania, Madagascar, South Africa, Senegal, and Greenland. So he gets around. So Adam spent around 2 years working on the Roseate Tern project for the RSPB, and he now lives in Aberfeldy with his family, where he runs the Aberfeldy Gallery. So Adam, the floor is yours. Welcome.
Right, thank you. I'm just going to try and share the right screen. Okay, here we go. Is that working? I'm not. I can't tell. I haven't got any controls anymore. Right. Sorry, I will just start. I haven't got any feedback. I've lost all of the chat. So I'm just going to assume that it's okay.
It's okay, Adam.
Great, thank you. So I'm going to try and keep up with the timings that I've already got. Most seabird breeding populations act as metapopulations. Individuals disperse between discrete colonies, which differ in their demographic rates. The demographic rates being productivity, survival, immigration, and emigration. Terns typically show higher rates of dispersal between colonies than other seabirds, moving to new colonies readily in response to disturbance or environmental change. The roseate tern metapopulation in Northwest Europe declined steeply from 3,812 pairs in 1968 to 561 pairs in 1987. This decline was put down to several factors at the breeding grounds, including human disturbance, depredation by gulls and rats, displacement from nesting sites by gulls, and high tides and coastal erosion. Trapping in the African wintering grounds was also thought to have contributed. Conservation measures, like we've been hearing about, were implemented at a number of sites in Northwest Europe to reverse the decline, including at Rockabill and Ladies Island Lake in Ireland, Cocotte Island in England, and several sites in Northern Ireland, Wales, and Brittany in France. These measures included discouraging gulls from nesting providing nesting terraces and nest boxes for roseate terns, and reducing human disturbance. Numbers have been recovering and reached 1,919 pairs by 2016. By far the biggest increase has occurred at Rockerbill. We studied the population dynamics of the main Northwest European colonies of roseate tern to inform the conservation strategy for this species in Europe. The objectives of the study were to estimate the demographic rates, productivity, survival, immigration and emigration at each colony, identify the demographic rates contributing most to temporal variance in population growth rate at each colony and of the whole metapopulation, and compare the population dynamics between the different colonies, assessing whether density dependence is limiting any of the demographic rates. We combined population counts, data on productivity, and capture, mark, recite data to construct a multi-state integrated population model, or more easily IPM, to help to understand the demographic drivers of variability in population growth rates over time. We used data from 1992 to 2016, which coincide with the period of recovery. This is when rich monitoring data began to be collected. We did not include the French sites in this model due to the sparseness of the capture, mark, recite data there. The populations of the 3 study colonies have all been increasing. Population growth rate has been quite steady at Rockerbill, but quite fluctuating at Ladies Island Lake and at Coquet. So these are violin plots, and these show the strength of correlation between the different demographic parameters and population growth rates at each colony. The main thing to note here is how important productivity and survival rates have been at Rockerbill, while immigration has been the biggest driver of population growth at Coquet, at least up until recently anyway. The correlations for Ladies Island Lake have been somewhat intermediate between the other 2 sites. These violin plots show the strength of correlation between productivity and survival at each colony and overall metapopulation growth. Perhaps unsurprisingly, survival and productivity at Rockerbill have been the main drivers of growth of the whole population, shown there with the yellow stars. Density-dependent processes occur when population growth rates are regulated by the density of a population. For example, productivity and survival may decline with increasing population density due to greater competition. Where density-dependent processes are observed within a colony, this suggests that there may not be much more room for population growth at that colony. These violin plots show the strength of correlations between demographic rates and colony size and are used to assess density dependence. The plots on the left-hand side are for survival and productivity. It appears that there is density-dependent regulation of juvenile survival and productivity at Rockerbill, but not the other 2 sites, where the centers of the violin plots for survival and productivity are close to 0. So the next 2 slides illustrate the movement of birds between colonies between years. The first is for juveniles. Most exchange involves Rockerbill, with Rockerbill a net source of young birds, which is indicated by the larger size of the arrows pointing away from Rockerbill than towards it. For adults, again, most exchange involves Rockerbill. Rates are lower, and this time rates to and from each pair of colonies is quite even. By using the model parameters, but then setting dispersal rates to 0, we can try to imagine how the metapopulation might have looked had there not been any dispersal. This is another way of looking at how dispersal rates have affected growth of each colony and also overall metapopulation size. In this figure, the solid lines with the red confidence limits show the model population sizes based on observed data. The dashed lines with the blue confidence limits show what is predicted to have happened with no dispersal between colonies, all else being equal. The model suggests that intercolony movement has restricted the growth of the metapopulation. At Rockville, the model estimates that without any emigration or immigration, the population would have over 900 additional pairs by now. For Coquet Island, without emigration and without immigration from other colonies, it would almost certainly have been in decline, with our modelling estimating that there would be around 90 fewer pairs by now. At Ladies Island Lake, there are likely to be an extra 50 or so pairs in the absence of dispersal. For the metapopulation as a whole, without the observed dispersal, model estimates that there would be around 860 more pairs than we find today. Clearly, this is mainly due to dispersal from Rockville, which has the highest productivity. However, this does not take account of density dependence, which could have been greater in the absence of movement. Indeed, we found evidence of density-dependent regulation of productivity and juvenile survival at Rockville, as we've been hearing about. More pre-breeders also disperse from Rockville than do established breeders. This may reflect density dependence too, where young, inexperienced birds may be better at other colonies where competition is less strong. Our data demonstrate that colony restoration is difficult. Even sites where management has greatly reduced depredation, nest site competition by gulls, greatly reduced human disturbance, and seen an increase in roseate terns, it could actually be sink sites dependent on immigration. Waiting for the next slide. Right. What's happened? It stopped. Sorry, it's completely stopped for some reason. I'm just going to try and sort this out. Here we go. Here we go. And the good news for our metapopulation is that juvenile survival at Coquet has increased in recent years, and very recently, after the current study, has seen a substantial increase in productivity, reaching 1.29 in 2019. So perhaps this site is now becoming another source population, less dependent on immigration. The continued presence of roseate terns on Coquet Island has bolstered the distribution of the species in the region and provides an attractive destination with lower competition for individuals dispersing from the Irish Sea colonies, mainly Rockville. The productivity of 1.29 seen at Coquet in 2019 is higher than at any time during the study period for this site, and also compares well with the mean values recorded at the other two sites during the study. Productivity at Rockville has been declining since 2009, which may be due to depredation of chicks by large gulls and increased competition for food by the increased numbers of breeding terns. The Rockville population is continuing to go up, although from what I've just seen, it looks like it might be panning out. However, if the productivity continues to go down, then this will eventually limit further growth of the roseate tern colony. It's hard to put a number on a specific productivity target for conservation managers to aim for, as population growth also depends on survival rates of the young birds and adults. Productivity at Coca and Ladies Island Lake as a mean over the study period were very similar, at just over 1. Possibly more significant for Coca's reliant on immigration was the low juvenile survival rate of 0.59 recorded between 1995 and 2010. However, annual survival rates didn't correlate between any pair of colonies. As we believe that the terns from the different study colonies winter in the same areas, factors in the wintering grounds do not appear to have strong impacts on annual variability in survival. Density dependence appears to be acting on productivity at Rockerbill. It seems likely that this will intensify. If this effect continues, then we are likely to see greater emigration of roseate terns from Rockerbill. If there are suitable sites available where the terns can experience good foraging and low predation, then these emigrants may achieve good productivity and survival rates, with overall net gains in the size of the metapopulation. If the possible destinations for terns emigrating from Rockerbill have either poor food availability or high predation levels, not to mention competition for nesting sites by gulls and disturbance, then increased density dependence at Rockerbill and a plateau in population growth there will also mean no further increase in the metapopulation. Site creation therefore seems crucial for continued metapopulation growth, as well as the security of an increase in the distribution and number of breeding colonies. When considering possible sites for roseate tern colonies, then an important first step is to look for established colonies of common terns, as these are apparently necessary for roseate tern colonization. Safety from most mammalian predators can be assessed quickly by looking at distance from the mainland shore. Then, studies of foraging success and, if possible, productivity of the common tern colonies can highlight those whose roseate tern restoration efforts are likely to be most successful. And if you want to see the full unabridged version, then the study was published in the Journal of Animal Ecology in 2018. And thank you very much for listening. Sorry about the little glitches.
Thank you very much, Adam. Thank you for wrestling with the technology, but you did very well, and what an elegant study that is. We've got a couple of questions, which I can just read out now. So from Richard Caldo, can we compare model population size predictions with observed colony growth rates to estimate the number of birds of breeding age that have not recruited to the 3 colonies yet and may be available to recruit to new colonies?
Yeah, I suppose so. I'm a little bit rusty on it all, but that sounds like something you'd be able to do. Yeah.
Okay. Norman has come in with a comment and a question. Outstanding work, Adam. Echo that. And he asks, what scope is there to model annual variability in relation to covariates like sea surface temperature or sardine stocks in Ghana, and maybe hindcast demography to understand past declines better?
Well, I suppose the beauty of this kind of modeling is that you can make them as complicated as you want. You can bring in more factors into play.
You've frozen on my screen.
Oh no, yeah, you froze as well, actually. It's probably my end. So I think the beauty of this kind of modeling is that if you can kind of visualize how it will work, how the actual system works, then you should be able to construct the model yourself. If you can get your head around the biology and how things would interact, you can probably recreate that in the model. So yeah, I think you could probably bring in these factors. So I think the danger is if you try to look at too many things, then you might end up with a complicated model that takes a very long time to run. you know, we're finding that things were taking days for one model run to complete. So that, you know, I think it's probably more of a logistical reason, logistical factors that might limit what you can do rather than, you know, the technical model itself.
Yeah. Okay, well, I think just incidentally on the sardine stocks in West Africa, we'll hear a little bit about this tomorrow, I think, but they are grossly overfished now on the Ghana coast, and there's extreme concern over the commercial sustainability of the fishery for the artisanal fishermen, far less the big offshore boys who are cleaning up the stock. So you might get a signal from that. It's obviously a very strong change going on down there. But
so what I could just say is that, you know, it'd be far more very satisfying to be able to bring in these kind of mechanisms into this kind of modeling, you know, really try and get a grip of what's going on.
Yeah. Okay, Adam, well, thank you very much. We're a little bit ahead of time, but it's been quite a long day, and so I don't think anyone will complain too much if we move on to our next presentation. I'm very excited about this because, you know, when I started looking at roseates 50 years ago, it was all pencils and string, but the technology has improved dramatically, and Chris Redfern is going to tell us about this in his presentation, Migration and Wintering Patterns of Roseate Terns Nesting in Great Britain and Ireland. And Chris qualified with an A-ringing permit in 1976, important thing to know that, while at Exeter University. He completed his PhD on mosquito cytology and endocrinology at Cambridge University. And after postdoc periods at the universities of Edinburgh and Liverpool, he ended up in an academic post in molecular and cellular biology at Newcastle University, where he's now emeritus, focusing on birds and ringing and migration interests and more. Forward to your presentation, and the floor is yours.
Okay, so
that's good.
Okay, so I got the point about recoveries from North America and Brazil, and that shows us that there's some potential for gene flow between both sides of the Atlantic. But although useful, ring recoveries are a rather blunt tool for migration studies, and we know very little about migration timing, refueling or staging areas, and how wintering areas are used. And now I know it's working because I can actually see myself on the screen too. Okay. Right, that's all changed now with the development of leg ring mounted geolocators or global location sensors. And these small cost-effective devices weigh just less than a gram when fully mounted and seem to be well tolerated by similar species such as Arctic terns. These devices record a number of different parameters, light levels, which is perhaps the most important for geolocation, but they'll also record temperature, emotion events, conductivity, and critically for geolocation, time, which we set to GMT. All those data have to be stored in an onboard memory. The battery should last a couple of years. The downside is, of course, we have to recapture the bird to obtain the data. Now, so the European Union Life Programme provided funding for geolocators, and we ended up carrying out a study over 2 years based at 2 sites. At Rockerbill in the Irish Sea in 2017, Steve Newton here on the right, and Paul Morrison, who you've already seen, we've seen both of them, fitted 20 geolocators to roseate terns trapped on the nest. And the following year, in 2018, they managed to recover 15 devices, and we were able to download data from 14 of those. And one key result was that there was no indication from the weights of the birds on recapture that the geolocators had had a detrimental effect on the birds. Following year, in 2018, on Coquet Island, North Sea, we were able to fit another 20 geolocators to roseate terns and got 16 of those back the following year. Of course, one nice feature of Coquet Island is that the colony is only about 1/10 of the size of the Rockabill colony. And that means that it's relatively easy for the resident assistant warden, David Kinchin-Smith, to keep tabs on individual birds and record when they were last seen and when they were first seen back on the island. And in addition, he was able to get detailed data on the breeding success of tagged and untagged birds. And that was very important because it provides further evidence about the effect of geolocators on the birds, or lack of. Another important aspect is that the Coquet and Rockabill colonies are linked because for many years the Coquet colony has been sustained and expanded by the immigration of birds from Rockabill, as you've seen from Adam's talk. So at the end of the 2019 breeding season, we had 30 sets of data, 14 from Rockabill and 16 from Coquet. So this next slide is a quick look at raw data. I always like putting this sort of thing in because it's really useful to see what the data look like. So this is just a raw light image plot from one of the birds. You see on the bottom axis we have date in individual days. The vertical axis is time during each day in GMT. And for each vertical line representing a day, there are varying densities of grey, and that indicates the light intensity. So we have nighttime up here, and bordering night, the red dots for the dawn threshold, light blue dots for the dusk threshold. And the darker blue line are the dawn and dusk thresholds for the tagging site, in this case Rockabill, throughout the year. So we can see that when there are departures of dawn and dusk or night length from of the geolocators from what we'd expect to see on the tagging site, that indicates the bird has moved. And you can see here there's a couple of clear movements to the west in autumn 2017 and again to the west in spring 2018. So this gives us a really nice indication that the birds have moved, and when we think about it, we can sort of work out what they're actually doing in a fairly broad way. Well, we can get a more sophisticated interpretation of these data using various packages, and we use one called FlightR, which applies fairly sophisticated methods to interpret where birds are from the light level data. And it also allows constraints based on lifestyle to be applied to improve estimates of geolocation. And for example, we might expect to constrain or give marine locations a higher probability for seabirds. So for the Rockabill and Coquet birds then, the migratory journey starts with the departure of the birds from the colony. And these colonies are shown with a red triangle for Coquet and a red square for Rockabill. I think you can see that most of the Coquet Island birds actually moved from the North Sea to the Irish Sea before moving south on departure. And that movement may well be over land because there are very few North Sea sightings around the English Channel. And I think most of the birds hung around in the Irish Sea or Celtic Seas before moving south on migration. The mean and median departure dates south were very similar for Rockabill birds in 2017 and Coquet in 2018, between the 10th and the 12th of September. Now, although the median departure dates were the same for the two groups, the birds in autumn 2018 had a rather greater spread of departure dates, as shown by these plots of latitude against date. So that's the 2018 data and 2017 above. So, and we think that these, the greater spread of departure dates is very likely a result of storms Ali and Bronner, which occurred in mid-September 2018, and they brought strong winds and heavy rain in from the west and were two of the most notable storm systems in recent decades at that time of year. One consequence of the analysis is that a number of birds in 2018 departed at around the time of the autumn equinox, and that made it hard to get a good analysis of the data for 2 of the birds using flight R. So what about the overall movement patterns? Well, they were pretty much as we would expect. So after moving south, all birds went to the Gulf of Guinea, moving down past the Iberian Peninsula, down past West Africa, and into the Gulf. The wintering areas are interesting. And as they're summarized on this slide for all the birds in the Gulf of Guinea. And all these points here represent wintering areas that the individual birds stay in for 50 days or more during the winter. So the central symbol are the mean coordinates of the wintering area. The circle is, or the diameter of the circle indicates is relative to the length of stay. And the vertical and horizontal bars give you an idea of the standard deviation of those locations. So of our sample of 30 birds then, 20 went exclusively to the coast of Ghana or due south, and you can see the heavy concentration of stationary periods around here. 3 of the birds went first to Ghana and then translocated partway through the winter and went off to Sierra Leone or Liberia exclusively. Sorry, or Liberia. 7 of the 30 birds went exclusively to Sierra Leone or Liberia. So 3 strategies of the wintering areas, and you can see those 3 strategies quite clearly in the light image plots if I've convinced you that they're worthwhile looking at. And perhaps the clearest one is this one, 51P4, and you can see that interruption partway through the winter where it's changed location quite clearly. And I think this is certainly quite an advance on what ring recoveries have told us in the past. So we know that most of our sample have gone to Ghana, but that nevertheless a significant, although smaller proportion, have gone to other areas of the Gulf of Guinea, particularly the Sierra Leone and Liberia part of the Gulf. So what about getting there? Well, this is a composite plot of all the stationary periods on the migratory journey. I think you can see the stationary periods, each of which is greater than 2 days long. There's a concentration in the Irish and Celtic seas. So there's some evidence for pre-migratory staging in these areas. There's also some birds stage in the Bay of Biscay and off the Iberian Peninsula, but the main staging areas for these birds are off the coast of West Africa, from the Canary Islands south to the border of Liberia and the Ivory Coast. The median duration of these stationary periods was about 4 days. And that's an important thing to keep in mind for later on. So the return journey then, coming back, the median departure date of the birds from the wintering areas is around 13th of April. Interestingly, the return trip was slower than the outward and took about 32 days, whereas the outward journey took about 18 and a half. And the staging periods on the return journey were slightly longer, with a median of 7 days, and that's shown on this graph in the center here. Most of the staging areas on the way back were off the coast of West Africa, from Sierra Leone north to Mauritania and Western Sahara. And there was particular concentrations of bird staging off southern Senegal and Guinea-Bissau. And the return speed also correlated with the departure date, so that the birds that left later in the spring were also in a hurry to get back to breed. As I said earlier, that these geolocators also record other environmental data, and perhaps the minimum temperature profiles are the most informative. And these show rapid transitions in temperature from the breeding areas to the wintering areas. Oh yes, so sorry. So these plots here are the data for the Rockabill and the Coca groups, and you can see that the temperature profiles are about the same. So we have this transition from the rather chilly British Isles waters to the rather warmer waters of the Gulf of Guinea, and you can see quite clearly the rapid outward journey, showing the increase in temperature, and the rather more leisurely return journey the following spring. So they confirm quite clearly the difference in migration speed. But what is also interesting about these data are the sections circled in green here. So on the return journey during spring, a lot of the birds have an extended period at intermediate temperatures on the way back. Now, for these minimum temperature measurements recorded by the geolocators, there's actually a very good correlation between satellite sea surface temperature measurements at the geolocations that we infer the birds to be. And that's shown by this graph here on the left. So there's a nice, very high correlation in temperature values. Now, when we look at a plot of the satellite sea surface temperature data shown here on the right, I've overlaid onto those the locations in blue of the birds within this circled area in blue on this temperature plot. Okay, so all the geolocation data for those time and temperature data are plotted here on this satellite sea surface temperature map. It's rather hard to see what's going on with these points. So if we take them away and you look at the plot without, and you can just about see there's a tongue of cooler water extends down from the Canary Islands along the coast of West Africa down to just below Guinea-Bissau. So this tongue of colder water actually represents the Canary Current. And the area corresponds to the Canary Current upwelling system, which is particularly prominent in spring. So what these data show is that the Canary Current large marine ecosystem is an important staging area for both the outward and the return migration of our roseate terns. The Canary Current large marine ecosystem is a major cold water upwelling boundary current, and it's economically important for fish, for local populations, as well as seabirds. And those fisheries are declining through overfishing, poor management and monitoring. And those habitats are also being degraded by overexploitation and pollution. And it does appear of the Canary Current marine ecosystem, this Senegalese upwelling influence of that, which is located in the area around Senegal and Guinea-Bissau, might be particularly important for roseate terns from the British Isles. With respect to the wintering areas of our birds also occur within upwelling areas, and those are all part of the Guinea Current large marine ecosystem. And within that, there are a number of subsystems, and two of those, the Sierra Leone Guinea Plateau subsystem, over here on the left, corresponding to the Sierra Leone Liberia part of the Gulf, and the Central West African upwellings are particularly important and coincide with the wintering areas of our roseate terns. These wintering areas too contain economically important fisheries, as Ewan says, and there's a great deal of legal and illegal fishing, overexploitation, and those fisheries may collapse within a few years if no action is taken to conserve them. So the conservation of those areas may be of vital importance for roseate terns and other seabirds. Those upwelling systems themselves, both within the Gulf and also the Canary Current, are driven by large-scale oceanographic and weather features, and they're also at risk from climate change. So just to summarize then, the Roseate Tern may be an upwelling specialist adapted to take advantage of these upwelling conditions. And it certainly seems that the upwelling systems of the Canary Current large marine ecosystem is an important migratory staging area. And the upwelling systems in the Gulf are also important wintering areas split between the Sierra Leone Guinea Plateau and the Central West African upwelling systems. And these areas may be important foci for exchanges between West and East North Atlantic populations and facilitate gene flow. We mustn't forget our local populations, and in the British Isles, local areas, and in the British Isles, the Celtic and the Irish seas are important as pre-migratory staging areas, and it's important too to look after the fishery stocks in those areas. And I'd finally like to thank everyone that's been involved in this project. The European Union Life Project provided all the funding via the RSPB. And huge thanks to Daniel Peach for organising the funding, motivating the team and channeling the money in our direction. And a huge thanks to David Kinchin-Smith for his observations and help on COCOT. And those have been key to helping us understand the effect of geolocators on birds. And thankfully, the effect seems to be so small that it's something that we can't measure with the current sample sizes. Also, I'd like to thank Steve for catching, tagging, and recovering, and his hospitality on Rockabill. Paul, of course, for catching on Rockabill and COCOT, encouragement, and for being the key motivator and organizer on the ground and at sea. And it was through him that we really got the project done. The geolocators were provided, we paid them for it, by Migrate Technology, but they kindly downloaded data from the thankfully few loggers that have stopped working during the winter. And finally, I'd also like to thank members of the International Roseate Tern community for their interest, insights, and ideas. So I think, any questions?
Thank you so much, Chris. That's fascinating work, transformative, really, I think, for our understanding of the flyway. We've never had those kinds of insights before at that granularity. So we have a few questions put in the box. So first of all, from Doug Radford, do birds from the Azores overwinter in the same places off Africa? And is there any exchange between birds from Britain and Ireland and the Azores?
Yes, there is some evidence. There are some birds from the Azores, I think, recovered in the Gulf of Guinea. And some of the birds that have been found breeding on the Azores have been recovered in South America as well. So yes, there is evidence that that interchange can occur.
Thank you. From Tony Murray, Ladies Island Lake is usually a week to 10 days behind Rockabill and I think COCOT. Sorry, I've skipped a question. Sorry, Alexandra Fink. Some COCOT Island birds seem to be moving towards Rockabill in the Irish Sea upon departure. Is anything known about the history of these birds, especially whether they originate from Rockabill?
Hi, Alex. That's a nice question. Yeah, right. So a lot of the birds that we tagged were already ringed, so we do know their origins, but there's nothing to suggest that the birds that we know came from Rockabill were any more likely to go to the Irish Sea than birds that we don't know their origins. So yes, it's a nice possibility, but there doesn't seem to be any evidence either way that that's a driver. Yeah, so it's an interesting question.
It's an interesting one. And just to say, I mean, I think there's increasing evidence from all sorts of seabirds that they cut corners. And I have a friend, Knockmigig, in the middle of Oxfordshire, and he had kittiwakes last spring over his house. And it's becoming more usual to discover these things. So, but it's a very interesting migratory short cut. So Tony asked, Ladies Island Lake is usually a week to 10 days behind Rockabill, and I think Coquet, you think Ladies Island Lake birds are leaving wintering grounds earlier?
I don't know. I don't know. They arrive back. The Rockabill and the Coquet birds arrived back at about the same time. I know they were different years, and they certainly left the wintering grounds at the same time. I would be surprised if there was any differences between colonies at this geographic scale.
Okay. And from Norman, excellent study, Chris. Do the immersion data on the loggers reveal roosting strategies at sea versus shore in Ghana? The counts of terns along the Ghana coast show a marked seasonal decline through the winter, and this was thought to be due to greater offshore roosting, or could be birds returning after dark.
Yeah, now that is an interesting question too. You would think that you would be able to answer it from the immersion data, but I don't think we can. So from in the wintering areas, then we've got two groups. We've got birds that winter off Ghana in the east and birds that winter in the west off Sierra Leone, Liberia. If you just ignore the birds that translocate partway through the winter. If we look at a number of parameters, say that the distance of those birds from the coast and also their nighttime immersion frequencies. Certainly the Sierra Leone Liberian birds are closer to the coast during the winter than the Ghanaian birds. And there is a difference in the nighttime immersion frequencies, so that the Ghanaian birds have a very high immersion frequencies, whereas the Sierra Leone Liberian birds don't. However, a big problem is that Ghana has quite a large salt industry. So there are a lot of coastal salt pans, and we know from historical observations that roseate terns, or terns in general in Ghana, a lot of them roost on these salt pans. So the thing is that with these geolocators, a bird roosting in a salty puddle would give you a high immersion count, and we wouldn't be able to distinguish that from a bird sitting on the water out at sea. So although we can use immersion data to show the behavior is different, we don't actually know why it's different. So we don't know where the birds are roosting in Sierra Leone, Liberia. Are they roosting on structures at sea, boats, or on the coast? I don't know. We'll really need to go to the wintering areas or use GPS loggers, something that has a much higher accuracy to find out where these birds are going in order to be able to answer that. So the immersion data are interesting, but in practice, they're really quite hard to interpret.
Yeah. Just to say, I've seen those roseates in the salt pans in Ghana, and they, of course, they use them and use those pans to a large degree, as you say, but they tend to roost on the little banks, between the pans. So it'd be interesting to see to what extent they actually contaminate themselves with salt water, but that's a good point. So Daniel Peake asks, would you agree that we could incorporate geolocators as a standard tool to monitoring environmental data along the migration and wintering grounds? What type of information would this give us?
Yeah, and I think it's an interesting question too. I think that there's a lot of potential for using geolocators in that way. They are now really quite small devices, but I think we probably ought to make them smaller. And I think technically we can do it. It just needs the will to do it and the funding to, package them smaller so that they're a lot less likely to have an effect. Although we've not had any detrimental effect in general on the roseate terns from these geolocators, they still look bulky. And any device that we attach to birds is going to increase the chance of accidents from environmental contaminants such as fishing lines and things that might get tangled on the ring and the geolocator. So I think yes, we should do that, but I think we should do it with smaller devices. So just to be absolutely certain that we're giving the birds the best chance they can.
Yeah. And I'm not sure I'll pronounce this name right. I'm sorry, Geoffroy Sitagetsi. Good presentation. Did you observe roseate terns staying all the year in wintering areas, failing to get back to the breeding site?
No, I'm not sure I understand the question. We can only tell that they get back when we've seen the geolocator and recovered the data. Is that? Yeah. So we have to get the birds back to know where they've been. Yeah.
Yeah.
And we've got back all the geolocators that were seen back on the breeding sites. So, and our return rate is about overall about 8%, but 16 out of 20 for Coca and 15 out of 20 for Rockabill. So yeah, the return rate is quite respectable.
Yeah. Okay, and John Atwood, finally, thoughts about geolocators versus GPS tags?
Right, I think the problem with GPS tags is that at the moment they need to be attached to birds with harnesses, and I don't think the evidence supports the use of harnesses on terns for year-round studies. It's yeah, people have tried it, and when people tried it, the return rates are very low, and I think it's possibly affecting survival. It's been done with Arctic terns, and survival rates, the return rates are very low. And I think with Arctics, we just don't understand what happens on the wintering ground with respect to their mass and things, and the harnesses may become too tight or constrain their movements. Interestingly, one of the projects we also have on the go is a colouring study of sandwich terns, and there was a report that actually came to me, not one of our birds, but of a sandwich tern that whose origin wasn't clear. And then it turned out that it was actually from a Dutch study, and that particular individual had a GPS attached to it by a harness. And when he looked at the photograph that the observer sent, and he saw it in Wales, the bird actually looked quite ruffled and the plumage was a bit disheveled. So I would worry greatly about using GPSs for year-round use on birds because of the harnesses. When they're sufficiently small to attach perhaps to rings or like geolocators, then we could do it, I think.
Yeah. Okay, Chris, well, we've two questions, so if we could have a brief answer as we're coming to end of time, but it's obviously generating a lot of interest. So Rebecca Jones asked, excellent study, thank you. Can the immersion data be used to look at feeding rates in different wintering areas?
Yes, I think so. I think we can do. That'll be daytime use. So we could get some information. I think probably it depends a lot on the programming. Sorry, that's not a brief answer, is it? But we would need to program the geolocators differently if we wanted to look in detail at feeding rates using immersion data.
Yeah.
But yes, it's a possibility.
Yeah. And finally, from Viv Booth, and this maybe speaks to Geoffroy's question, have any birds with geolocators been seen subsequently, i.e. had a year off from breeding or been elsewhere, then gone back to the site where they were tagged?
Not for the roseates, as far as we know. For Arctics, that's a different matter. But slightly different variants on that. One of the Rockabill geolocator birds that we know from a geolocator was in an area of just south of Senegal on the return journey, was actually trapped by ringers in Senegal 2019, I think. So that bird, and that's rather nice because it shows that individual is using the same area in different years. So a correlation of geolocator data with other observations. So yeah.
Well, thank you very much, Chris. And if I chair's prerogative, just one very brief comment. You're finding that the distribution in the winter is further up the west coast than just Ghana. I think is really important, because especially as those birds are closer inshore, because you know that tern trapping is continuing in different countries like Sierra Leone and Mauritania. So that's something for us all to think about, I think.
Yeah.
So thank you very much, Chris.
Thank you.
Brilliant presentation. And we're now going on to our final talk of the day from Richard Berridge, visual tracking of roseate terns from Rockabill. Richard has a background in the management and monitoring of seabird and tern colonies as a warden. And for the past decade, he's been heavily involved in ornithological monitoring and research, primarily at offshore wind farms in his role at Econ Ecological Consultancy. Visually tracking sandwich terns to assess their response to a wind farm over a period of 6 breeding seasons has led to further projects focused on Arctic and, as presented here, roseate terns. So, Richard, over to you.
As introduced, I'm Richard Berridge. I'm going to present an overview of a project we worked on in the summer of 2018 out at Rockabill, visually tracking roseate terns. Obviously, this isn't an individual effort. Martin Perrow and Andrew Howard were heavily involved as well. So the visual tern tracking technique has been developed over a number of years now. This picture from Rockabill shows that we can really conveniently sit right alongside the colony. We're close enough here to read even the numbers on the tern boxes. So we have a good idea of which birds we're picking up to track. We'll identify a bird leaving and then follow it. I'm afraid my screen share is flying through this much quicker than I might like. So this is what the raw data looks like. We take waypoints at every behavioral observation or association with a feature. You can see it turns into a little bit of a scroll, but I promise you that all of those codes and abbreviations do mean something. So just to give you a better idea of what this actually looks like, I'm going to run a short video, which I hope works. The video is actually us tracking sandwich terns through an offshore wind farm. So you can see it can be quite a challenging technique. It is pretty hard on boats. At Rockabill, we actually went through a couple of boats before finding something suitable. Initially, we were on a large 12-meter cabin rib. This boat was simply too slow. We replaced that with a 6-meter rib with 150 horsepower outboard. It was quick enough, but we broke it in fairly short order. Eventually, we settled on an 8.5-meter rib, which was quick enough and sturdy enough. The boats themselves obviously aren't enough. You need an expert skipper, not only to keep you safe, but also to actually follow the birds, which is no mean feat. And of course, we also need some hardy and experienced observers to actually collect some data while we're getting thrown around out there. This survey technique, while being tremendous fun for those of us with a certain mindset, is maybe not for everybody. So in terms of the tracking statistics, what we actually managed to get done, we did 11 days of tracking, 89 birds tracked. 61 of those were completed foraging trips, that is out from the colony and back again. Roseate terns big fans of gregarious bathing just offshore of the colony, so most of our aborted trips were birds that were simply leaving to have a wash. Of these foraging trips, we managed to complete about 51% of them. 31 are completed. 30 are incomplete. Our most common reasons for losing a bird would be either in a large feeding aggregation, we'd simply confuse it for other terns. So looking at our tracks plotted here, due to the boat issues, we were in 3 different time periods in different colours. A total tracking time of just over 40 hours. You can note on the plot that our earlier trips in June and early July are more coastally focused. Later in July, mid-late July, some roseate tern chicks were actually fledging by the time we left. You can see a much bigger outward fan from the island into deeper waters. So there's some clear temporal differences there across the breeding season. Our mean track time is 39.6 minutes. A max time of nearly 180 minutes, so quite a significant time to be following a bird in a boat. Our total track distance, 1,288 kilometers, mean distance of 21.1. A maximum distance of the colony of nearly 30 kilometers. You can see a mean distance of just 8.5. And we found roseate terns to be surprisingly fast. The speeds here actually compare fairly favourably to sandwich tern, which are obviously much bigger and more powerful than roseate terns. A max speed of nearly 57 kilometers an hour is fairly impressive. So some tracks and trip statistics. In terms of flight heights, you can see that just 83% of the time is spent at relatively low heights, 1 to 15 meters. Just 2% of time at greater than 20 meters. Commuting flights were generally of less than 5 meters. We observed fishing from heights up to 25 meters, but 96% of our attempts observed were from less than 15 meters. And 52% of attempts were from that very low flight height band of just 1 to 5 meters. We actually, so some example tracks, and just a few cherry picked here to show the level of data that's being recorded. Different tracks in different colors, obviously, and then foraging attempts overlaid as black dots. Mixed species foraging associations are the yellow dots. Groups of auks in green. Some images here just to show you what that might look like. There can be some really significant feeding aggregations out there in the Irish Sea, multi-species. There's also the next layer of data that we can record, so associations with seaweed, for example, or an instance here where a bird lost a fish to a skewer. We also have mixing fronts, note of prey being carried back to the colony, for example, local fishing boats operating in the area. So looking in depth at some foraging, we found that roseate tern forage in a range of habitats, including really quite deep water in the central Irish Sea. You can see a clear cluster around the colony as well. All of these plots are different prey species being taken from different areas. Black are obviously the tracks of the birds themselves. So in terms of foraging rates, completed attempts per hour, a mean of 36.6 with quite a large standard deviation around it, and a max of nearly 210 attempts per hour. Roseate terns tend to abort a lot of attempts as well, nearly 40%. So that would involve a bird starting a dive and then pulling out at the last minute, for example. So as mentioned earlier by Steve, roseate terns do attend these mixed species foraging aggregations, which are usually driven by auks pushing prey to the surface. But roseate terns appear to be quite unsuccessful in taking advantage of any opportunities arising. They're probably pushed out by the bigger, more powerful species. Kittiwake, herring gull here, gannet at the surface. So they tend to leave these aggregations quite quickly. But you can see from the plots on the right just how frequently they may attend these kinds of aggregations. It can often feel like the tern is leading you from one aggregation to the next while not catching any fish in doing so. So we assume they're probably looking for an early opportunity when the auks first start to drive a bait ball up to the surface. So looking at the prey at sea, we find a fairly classic pattern where most of the prey consumed by roseate terns at sea tend to be very small and are therefore usually unidentified. The graph here shows a percentage of prey items as individuals, but also by biomass below. You can see in the consumed portion, lots of, running away from me again, sorry. So we have a consumed, a carried and a provisioned portion. This graph really just shows you nicely how the consumed portion tends to be rather a lot smaller than the carried portion. Obviously, bigger items are more valuable to a chick, so these will be worth the energy required to carry them back. And we can see that clipeids and sand eels tend to dominate the chick provisions. These are high energy items. Roseate terns are also taking small invertebrates and such like, which are obviously less energetic. So looking at prey delivery, we observed 28 prey items being carried back to the colony. This was over the entire foraging range. We can see a prey item carried back from very nearly the full 30 kilometers that we tracked birds to, but most do appear to be fairly well clustered. But it's clear that roseate terns appear to operate a little more like a sandwich tern and less like Arctic terns, at least Arctic terns at skerries on the other side of the Irish Sea, which almost exclusively forage very close to the colony. Of course, this can come at a cost, as mentioned previously. The further you have to carry a prey item for, the higher risk you are of losing it to a kleptoparasite, for example. So, looking at some provisioning data provided by Birdwatch Ireland, obviously Steve has presented some more up-to-date data than this. This is from 2016, 17 and 18 inclusive. The data is taken from some fairly heroic whole day, up to 17-hour watches undertaken by the wardens. These watches will focus on a group of nests could range from 18 to 27 nests in these cases. There's a known number of chicks. And we can see from this there's some annual variability, but clipeids are generally very important in terms of provisioning chicks on Rockerville. So in terms of calculating biomass, the data on the bottom of this graph is biomass, above is simply the individuals. Calculating the biomass of a prey item relies on an accurate sizing of that item, which we do by relating it to bill length. So looking at estimates from our tracking data compared to the colony data, we can see that items tend to look to be smaller in the colony data compared to what we might expect from what we were observing during tracking. That's for clipeid above and sand eel below, you have a similar pattern. So we have a low sample of tracked birds carrying prey items compared to the observations by the wardens. We have just 28 items. compared to their 1,070 in 2017, for example. However, tracked birds' prey items are supported by multiple photographs, so we would tend to try and race ahead of a tracked bird as it approaches the colony, slow the boat right down to keep stable, and then Martin would very skillfully fire off some photographs as the bird flew past us. So very useful for us to be able to calibrate our observations against the photographs. Of course, there's always a slightly competitive element when you're estimating prey limb sizes, which we can then ascertain just how accurate we are. So images here just to show the difficulties, not only in identification, but also in assessing a size relative to bill length. These two images in the bottom right, it's the same bird and the same fish, but obviously they look rather different depending on that angle. So obviously we were intrigued as to what these clypeids are. There had been a historical suggestion that sprat were more frequent around Rockerbill. But certainly in the year that we were tracking, the general colour of the fish we were observing, the body shape, and the position of the root of the dorsal fin relative to the pelvic fin suggested we were mostly seeing Atlantic herring and not as many sprat. So, assuming that the herring are key, it's interesting to look at where they're spawning, and the Morn spawning ground is just to the north of Rockerbill, and we could assume that that might be fairly important in terms of the ongoing success of the colony there. There's also a spawning ground off the Isle of Man. Both of these likely to be important not just to Rockerbill, of course, but other seabird colonies throughout the Irish Sea, including Lambay Island just to the south of Rockerbill. So looking at our data in terms of the current SPA and a potential extension of it, the current SPA is this black box. It's 3.5 kilometers squared. And it's based on the distribution of roseate terns from some historic boat-based survey data. This box can actually be seen to do a fairly good job of delineating an important core area for roseate terns. Processing our tracking data through the BirdLife IBA script, we think we could make a fairly strong case, especially using the KDE feeding fixes for foraging birds, of at least a 20% boundary marked out here in red, but possibly even out to 30 kilometers, which incidentally is the range of our tracks. So in conclusion, visual tracking has considerably increased our knowledge base on spatial utilization and the interactions of roseate terns with their prey and other species. The tracking was really nicely complemented by the colony studies, the data from the wardens on the island. The visual tracking method is non-invasive. Obviously, there's a great deal of concern about just how easily disturbed roseate terns are. We've confirmed a foraging range of up to 30 kilometers. Of course, it may be bigger in some years, it may be smaller in others. We now believe that understanding common tern, both in its own right and in relation to roseates at Rockerbill, might be essential to furthering our understanding. Quite an interesting point in relation to Steve's musings about sandwich tern as well at other colonies earlier. And finally, it's likely that the Irish Sea herring stock could be critical to most, if not all, tern species at most, if not all, colonies. And again, just looking back at our potential 20-kilometer buffer for an important area for sandwich tern, it's worth noting that the spawning ground for herring is actually outside of that protected area. Could be a further consideration. So just to give some thanks and acknowledgements, obviously the RSPB for funding this work through the EU Life project, and the collaboration with Birdwatch Ireland, all the help they've given us. And of course, all the drivers, fixers, friends we've made and colleagues at Rockerbill and Skerries on the mainland. And if you're interested and haven't read it already, most of what I've presented here is presented in more detail in a British Birds paper published last year. And with that, I will stop sharing my screen, start my video, and answer any questions.
Thank you very much, Chris. Can you hear me okay?
I can hear you, Ewan, yes.
Yeah, we haven't got any questions in the box, so I would just like to ask you a couple of things and reflect on a couple of things. My screen froze for a few seconds when you were talking about the size of the fish carried being larger than the size provisioned.
Right.
Can you just tell me what you think's going on there?
Sorry, could you repeat size of the fish carried?
At one point, I think you said the size, the histogram showed the size of the fish carried was larger than the size being provisioned.
So the fish from the colony data were tending to be recorded as smaller as the fish that we noted as being carried.
Yeah.
Is that what you mean?
Yes. So is that an observer? Do you think that's an observer bias?
I believe so, yes.
Yeah.
So going back to the point about we tend to try and photograph whatever we can, because it's incredibly difficult to assess how big a 5 centimetre sand eel is when a bird obviously lands and rather promptly passes it to a chick. The fish tends to be bent across the bill. It's an incredibly difficult thing to do. And I think the point really is that in order to do it well requires quite a lot of experience, quite a lot of training, and preferably quite a lot of ground truthing through photography.
Yeah.
Which is really the point we're trying to get across. Not necessarily that anybody's doing it wrong, just that these things could often be done better.
Right. So, okay, Richard, well, a few questions coming in now. How did you estimate flight heights?
Visually, but we also use a very small handheld laser rangefinder, which is actually designed more for forestry work, or golfers are also fans of them for figuring out distances to tees and so forth. So this small handheld unit with quite a high degree of user aptitude can be used to, for want of a better word, ping a tern in flight. It then triangulates and gives you a height above your eye height, which obviously is measurable. So we can verify our visual observations. We've done this not just through tracking, but also on boat-based surveys on bigger survey platforms to attempt to see just how accurate we are. We tend to be fairly good at it. If anything, people tend to underestimate, but obviously with Roseate Tern tending to fly relatively low heights, it's fairly easy to estimate a flight height of 5 meters or 10 meters. Obviously the 5 meter bands makes it a little bit easier. Estimating very high flight heights is obviously very difficult, especially with no frame of reference. If you're in a wind farm, for example, you're surrounded by reference points because you know the heights to specific structural parts of a turbine. But in open ocean, you've got nothing but a hunch.
Yeah.
But over the years, that you can build up a pretty clear picture, especially by using this laser rangefinder to ground truth yourself and start to inform your estimates.
Great. Good. Well, the questions are coming in thick and fast now, so see if we can get through in the next 5 minutes. So if we can be brief. So the provisioning monitoring is done on land on 3 days through the season. Were the time periods the same for the boat-based observations? The data seem to have been combined in the graph. That's from Viv.
I believe that one of their, I mean, Steve might be a better well, Steve is a better person to answer this than me. But I believe that the free provisioning watches are on set dates and they're repeated annually. Steve's very kindly logged in. And from memory, we may have been tracking, we certainly were on island when the guys were doing one of those watches. So I believe actually, yes, one of our tracking days coincided with one of those observer days. Steve may know or not remember. It was a couple of years ago now.
Yeah, you're quite right. No, yeah, you were right. One day would coincide with our third assessment in that mid-July period. That they're not fixed dates. They're relative to the breeding season. So we're trying to time them, say, on, the first few days after the first chick appeared, then what we call the middle chick period when they're about a week, 10 days old. And then our last one, that one in 2018 was in mid-July, was when the bulk of chicks are fairly large and indeed a few start fledging. So we're trying to pick up different phases of chick development through that. But it's very varied with the first egg dates and the timing of the season, which can possibly shift around about a week.
Okay, Steve, thank you very much for that clarification. I'll stop you there because let's try and squeeze in a couple of quick ones. Yep. You say Richard, you say the visual tracking is non-invasive. How is this measured? That's from David Kinchin-Smith.
So when I say non-invasive, I simply mean that we're not going into a colony, disturbing that colony and handling birds. It's totally hands-off. We continually monitor for any evidence that we're distracting or disturbing a bird in any way. And it's quite incredible that you can be extremely close to them and they are not bothered. They will, Roseate Tern included, all the terns, will take a fish from literally right next to your boat. They're not fussed about you at all. So it's simply in comparison to other popular methods which often involve having to capture a bird.
Okay, and final question. I think there's two more, but I think we'll have to respond to those out offline. From Norman, why use a radius as the recommended SPA when the isopleths provide a more parsimonious protected area? The radius includes a lot of sea that the birds never used.
Yeah, the radius was really just to illustrate a point. Obviously, we don't have an awful lot of data. There's every reason to believe that it would be possible in other years that those areas which we didn't observe birds in actually hold birds. Maybe at other times in the season, it all depends on where the prey fish are. So I don't think we would want to suggest a new SPA based on what we have. It was just an interesting exercise to have a look at our data in contrast to what there is existing currently.
Okay. Well, you're very popular, Richard. We've got 4 or 5 more questions, but I'm afraid I'm going to have to park those in the interest of time. But thanks so much for a really interesting presentation. I really loved that.
Good. I'll answer the questions by text. I can do it now, I think.
Okay. Right. Well, that's the end of this session. In fact, the end of day 1. I'd like to thank all our speakers. Really excellent presentations and such innovative and elegant work, all 3. And indeed, I'd like to thank all the presenters today for an excellent first day. I'm sorry about the few technological gremlins to begin with, but this is all very cutting edge stuff. I think I would also just like to thank all the participants. I think we had up to 80 this morning, so very well done if you managed to hold the course throughout the day.